Have you ever actually stopped to think about the incredible biological factory operating inside you right this very second? I mean, really think about it. Every single time you get a paper cut or a scrape, this massive, hyper-coordinated manufacturing process kicks into gear literally just to save your life. Today, we're exploring the hidden blueprint of blood clotting, specifically looking at the incredibly dynamic partnership between two absolutely vital proteins, Factor VIII and von Willebrand factor.
It's kind of like a giant biological puzzle, right? How exactly does your body manufacture the perfect molecular glue to stop bleeding? It has to be sticky enough to plug a leak, for sure, but strictly regulated enough so it doesn't just clog up your entire circulatory system. Well, the answer lies in a highly specific problem-solution framework that happens at a microscopic level the second you experience any vascular injury. Okay, let's dive into this. Here's our roadmap. 1. The genetic blueprints. 2. The cellular protein assembly lines. 3. A look inside of von Willebrand factor. And finally, number 4, the ultimate clotting partnership.
All right, kicking things off with Section 1, the genetic blueprints, distinct origins. Think of every great buddy cop duo. They always start out with completely different backgrounds, right? Well, zooming right into the nucleus of endothelial cells and megakaryocytes, we find ground zero for our molecular factories. And here, we can trace two completely separate origin stories. On one track, we have the beginnings of Factor VIII, and running parallel on an entirely different genetic pathway, we find human von Willebrand factor.
What's honestly amazing here is just how genetically distant these two partners are before they finally meet up. Factor VIII, it's built from a blueprint way over on the X chromosome, specifically at position Q28, and it's a 100 kilobase gene. On the flip side, von Willebrand factor's instructions are housed on chromosome 12p11.3, and it's a significantly larger blueprint, a 190 kilobase gene spread across 52 exons. So they start their lives on completely different chromosomes, but their ultimate destiny is to work together. Moving right along to Section 2, cellular protein assembly lines, building the pieces. So now that we have the genetic blueprints, let's step onto the factory floor. Our cells basically operate like rigorous, step-by-step manufacturing lines to build these complex proteins. Following the assembly line for Factor VIII, it's an elegant, relatively straightforward process. It kicks off with mRNA translation from a 9kb poly-A, which gives us a single chain made up of 2,352 amino acids. Next up, a signal peptide is cleanly cleaved off. Finally, the protein folds itself into its mature shape and is secreted out of the cell, ready for action. Pretty efficient, right? Meanwhile, building von Willebrand factor, that requires a lot more heavy lifting. Step 1 begins as pre-pro VWF translation, but then it has to connect with itself in a process called C-terminal dimerization, creating a pro-VWF dimer. The assembly doesn't stop there, though. In Step 3, through N-terminal multimerization, these dimers vigorously link together to form massive, sprawling chains. Finally, in Step 4, a biological tool called furin acts like a pair of molecular scissors to cleave off the propeptide, giving us our mature VWF multimers. Now, because these VWF multimers end up being so unbelievably huge and complex, the endothelial cell actually needs a special shipping container just to hold them all. Enter the Weibel-Palade body. This is a highly specific cellular storage granule that houses the newly assembled von Willebrand factor. It holds it perfectly primed and ready for regulated secretion at the exact second your body needs it to stop a bleed. And when I say these structures are large, I am not kidding. The final VWF multimers range in mass from 0.6 all the way up to a staggering 20,000 kilodaltons. This massive scale isn't just some accident of biology. It is exactly what makes VWF the perfect biological net to catch cells and physically stop a bleed. Okay, Section 3. Inside von Willebrand factor, form meets function. Let's shift gears and zoom in even closer. We know VWF acts as a massive biological net, but what does its actual molecular anatomy look like? Now, what's really interesting about this is when we break down the primary VWF translation product into individual functional zones, or domains, it actually looks a bit like a giant biological multi-tool. Just like a Swiss Army knife has a different attachment for a different job, VWF has distinctly tailored segments. If you look closely right at the middle section, we find specific, highly specialized domains labeled A1, A2, and A3. Let's break this down to make it super easy to digest. We can basically map out these sticky points. The A1 domain, it's a major multitasker, binding to GPIB, heparin, and collagen. The A3 domain is specifically dedicated just to binding collagen. And crucially for our story today, the D and D3 domains are perfectly shaped to bind to our other hero, Factor VIII. This brilliantly illustrates how every single part of the VWF molecule has a highly specific job in the broader clotting process.
But here's the catch. A net this massive absolutely has to be carefully managed. That's why regulatory cut points are so vital. As we noted earlier, furin is the enzyme responsible for removing the propeptide either right before or soon after secretion. But there's another crucial set of molecular scissors, ADAM-TS13. This enzyme specifically targets and cleaves within the A2 domain. This step is absolutely essential for breaking down those massive 20,000 kilodalton multimers. If it didn't do this, they would just spiral completely out of control and cause unwanted blockages in your blood vessels. And understanding this physical structure explains a whole lot about real-world medical variants. When there's a defect in the VWF gene, it behaves completely differently depending on where exactly it happens. For instance, type 2 genetic variants cluster precisely in those highly functional domains we just talked about, creating patterns like 2A, 2N, 2B, or 2M. This is completely different from type 1 and type 3 variants, which are distributed seemingly randomly throughout the entire molecule. Simply put, when you break a specific domain, you break a specific function.
Which brings us to our grand finale, Section 4, the ultimate clotting partnership, teamwork in the plasma. So we've built our proteins, we've explored their individual structures, and now it's time to zoom back out. Distilling this whole explainer down, von Willebrand factor essentially performs two essential jobs to keep us alive. First, it acts as an anchor, using its adhesive binding to grab onto matrix molecules like collagen at the exact site of a vascular injury, and then it rapidly captures passing platelets to form the primary plug. Second, and honestly just as importantly, it acts as a protector, completely stabilizing Factor VIII while it travels through the circulation. Finally, out in the bloodstream, we see this incredible biological convergence actually happen. The massive VWF chain literally grabs onto the much smaller Factor VIII protein. VWF anchors it, physically protects it from being degraded too quickly, and delivers it precisely where it needs to go to form a stable, life-saving clot. It's just an incredibly beautiful, highly synchronized biological dance. But it does leave us with a pretty provocative thought. Knowing how precisely these domains interlock, if just a single genetic variant in the A1 or A2 domain alters the shape of this molecular glue, how might it unravel this entire delicate partnership? It's a stark reminder of how microscopic changes in our genetic blueprint can ripple out to affect our health.